Endoplasmic reticulum was discovered by scientist called as Porter, and the name endoplasmic reticulum was also been given by the same scientist K.R. Porter. So here endoplasmic reticulum are single membrane structures, single membrane cell organelles which are spreading throughout the cytoplasm, and these are concerned, or we can say that these are structures which are extending between outer membrane of nucleus to plasma membrane. So that means that they are spreading throughout the cytoplasmic matrix, and these are the structures which are made up of lipoproteins. So majority of the lipoproteins which are present in the cell are existing in the form of endoplasmic reticulum. And these endoplasmic reticulum, if you consider the diagram, so here this is a nuclear membrane. Consider this as a nuclear membrane, that is nucleus here. So here we can see the extensions which are coming like this. So this type of extensions which are coming, so this is called as endoplasmic reticulum, and this structure will be extending up to the plasma membrane, so which is going up to the plasma membrane like this. Then on other side also, on all the sides this type of extensions can be seen, and here these are this part which is present inside the endoplasmic reticulum, we call this as a luminar part, and outside the endoplasmic reticulum the cytoplasmic contents are there. No, this is called as a extraluminar part. Outside, this part which is present outside is called as the extraluminar part, and the luminar part is filled with amorphous substance, and here this is a single membrane. So this single membrane itself is showing the folding like structures, and the endoplasmic reticulum is made up of three parts actually. So those three parts are vesicles, tubules and cisternae. The parts are cisternae. Cisternae are elongated structures which are unbranched, and these are the structures which are arranged parallel to each other. So this type of structures are called as cisternae. And tubules. Tubules are also long and elongated structures. Sometimes they may be branched or sometimes they may be unbranched also. This type of structures are called as tubules. Whereas vesicles. Vesicles are generally spherical shaped structures. There is a lot of change in the size or volume of these vesicles, which are ranging from 25 to 500 nanometers. Whereas tubules are ranging from 40 to 100 nanometers. So tubules are showing a measurement of 50 to 100 nanometers in the thickness, whereas cisternae are showing 40 to 50 nanometers in their thickness. This type of variation can be seen in the sizes, and one endoplasmic reticulum is made up of all these three structures. So which is a combination of all these three parts, and here endoplasmic reticulum will exist in two forms. Those are called smooth endoplasmic reticulum and the rough endoplasmic reticulum. First, the rough endoplasmic reticulum. Rough endoplasmic reticulum are the membranous structures which are studded with the ribosomes on their outer surface. Consider this is rough endoplasmic reticulum, where on outer surface they are studded with the granular structures. These granular structures are called as ribosomes, like this. So as granules are present, otherwise we can also call it as a granular endoplasmic reticulum. So it is having a rough texture because of the granules, and at the same time, so we can also see some second type is called as a smooth endoplasmic reticulum. Here, this from here to here where the granules or the ribosomes are studded, so we call it as a granular, and so from this region to up to this region, the ribosomes are not studded on the outer surface. So the texture of this part will be smooth. So we call it a smooth endoplasmic reticulum, or otherwise agranular endoplasmic reticulum. Here that means rough endoplasmic reticulum and the smooth endoplasmic reticulum are continuous with each other. And as we are saying, the ribosomes are present. Which type of ribosomes are there? As these are the eukaryotic organisms, the ribosomes are of ATS type. ATS type of ribosomes are studded on the outer surface of the endoplasmic reticulum that forms a rough endoplasmic reticulum. So here either it may be a smooth or the rough endoplasmic reticulum. Both of them are concerned with the spreading of the materials, and these are acting as a, and particularly endoplasmic reticulum is acting as a intracellular transportation channel. Endoplasmic reticulum is acting as a intracellular transportation channels. So here intracellular means within the cell they are helping in transportation of materials from one side to the other cell. How they are helping in? Actually these endoplasmic reticulum are concerned with the formation of vesicles. Though the vesicles are nothing but some material is packed inside the vesicle, and that vesicles will move from one side of the cell to the other side of the cell. So vesicles are helping in the transportation. So that's why transportation within the cell, that's why we call it as the intracellular transportation channels. Then here if you see the functions of endoplasmic reticulum, the functions of endoplasmic reticulum, so these are the structures which are concerned with the formation of vesicles like structures, and also in the formation of Golgi complex. The vesicles of the Golgi complex are formed, or the Golgi complex will arise from the endoplasmic reticulum, and endoplasmic reticulum along with the Golgi complex, both of them will form the vesicle like structures which are helping in the formation of lysosomes. As we know that lysosomes are the spherical shaped structures which are having a specialized enzyme called as hydrolytic enzyme concerned with the digestion. Okay. Then smooth endoplasmic reticulum, so these are concerned with the synthesis of lipids, and particularly in the animal cells these are concerned with the synthesis of steroids. Then rough endoplasmic reticulum, as this rough endoplasmic reticulum is studded with the ribosomes are concerned with protein synthesis. Actually the place where the ribosomes are attached on the outer surface of endoplasmic reticulum, there some perforations are present. Through that perforations, whatever the proteins that have been synthesized, those proteins will enter into the luminous part of the endoplasmic reticulum, where they are moving from they are converted into packaging materials, and from there that is converted into vesicles, and from there the protein part will be transported from one side of the cell to other side of the cell. Likewise these are helping in transportation also within a cell. Rough endoplasmic reticulum contains glycoproteins. So those are ribofourin 1 and ribofourin 2. So these are the special type of proteins which are helping in attachment of ribosomes to the outer surface of endoplasmic reticulum. And if you see the other functions related to the endoplasmic reticulum, endoplasmic reticulum is also being seen in the plasmodesmata region. Wherever the plasmodesmata is present, endoplasmic reticulum is present, that part is called as desmotubule. Or simply to say, the plasmodesmata contains endoplasmic reticulum, that is called as a desmotubule, and because of this desmotubular structure, it will help in transportation between adjacent cells also. Transportation of various materials between adjacent cells also, and the endoplasmic reticulum, as it is forming into a cytoskeleton structure inside a cell, so it will also help in the maintain the position of cell organelles. position of the cell organs will be remain constant and stable, nearly stable, due to the presence of endoplasmic reticulum. So this type of this type of lipoproteinous membrane structure which is concerned with the transportation is called as a endoplasmic reticulum, which exists in two forms, and that those are rough endoplasmic reticulum and the smooth endoplasmic reticulum, and those structures are made up of three parts: cisternate, tubules and the vesicles. And the vesicles which have been formed, as I said, that they are helping in the formation of lysosomes and sometimes in the packaging of materials and transportation of the protein like substances or the other materials from one end of the cell to the other end of the cell.